So after around 8 billion years from when the universe was formed, around like if you look at the time, like around 4.6 billion years ago from now, the sun was formed. And shortly after that, around 4.5 billion years ago from now, the earth was formed. But then a miracle happened. Then we saw life at around 3.7 billion years ago. And this is a real miracle because we still do not know how life evolved. There are lots of theories. There are theories of panspermia. There are primordial soup. There are a lot of theories, and I'm not going to go into that. But what I was interested when I started doing my PhD, I was interested at looking at life in deep dark biosphere. So when I was given this task, I was overwhelmed because when you start looking for something, even in space or in any other way, you need to define it, right? So what is life? So you need to find a definition for it. So suppose somebody asks you to look for a car, a red car, you should know that it has got four wheels, some model number. But when somebody says you like go and look for life, you need to find a definition. So what I started doing is that I started looking for the answer, what is life? A very common thing, we look for life in extraterrestrial planet, we look for life in different places on our planet Earth in extreme environments. But what is life? So then I came up with some of very confident theories, I would say, by different scientists, different literature. So some say life is self-organized. So it means that it's in this equilibrium with the environment. So it gets organized by itself. But there was a chemist in the room where this talk was going on, said that there are chemical reactions which can self-organize by itself. So it's not a characteristics of life. Then certain biologists came together and they told that evolution and reproduction are part of life. So these are intrinsic traits which you see in any living beings. So then somebody said that what about the male bee workers who cannot reproduce in a community of bees? Aren't they alive? Or what about a sterile organism which cannot reproduce? An adolescent boy, like they cannot reproduce, right? So aren't they living being, right? And evolve, if you think about evolution, like a lot of crystals can evolve their structure when they're exposed to certain conditions. They can also replicate, right? So are they life? If you think about fire, like it can replicate on its own. Like if it gets the proper thing, like it can move from one to another. Then I was in like swimming in a big ocean. I didn't know what. Then in 2012, just 1 or 2 years before starting my PhD, somebody published a paper and they told that life deficiencies have wasted a lot of time, energy, and money that could have been used for more useful projects. And I took that as a Bible, so I started following that. I thought, no, we will look at life, but we will look at life that we know, the simplest form of life. Those are the microbiome or microbes, microorganisms, which we cannot see with our bare eyes. So some statistics regarding the microbes on our planet. So number of microbial cells on our planet is much greater than number of planets in our galaxy. So we have got a huge diversity and huge number of population of microbes on our planet. And at the same time, we do not know 99% of those microbes. And even those 1% that we know, we cannot culture it, grow them in our lab, and look at their physiology and other characteristics. So it's still an unknown realm. But you will be interested to know that most of the microbial life form on our planet are in the subsurface environment. In 1998, a phenomenal paper called Prokaryotes, the Unseen Majority, was published. And Whitman et al. Published that most of the microbial cells are buried beneath the surface of the earth. And before that, there were studies where people thought that life couldn't survive beneath tens of meters from the surface. It can survive in soil, different layer, but not beyond that. In 2018, it became much more evident that life can survive in the subsurface. And if you think about prokaryotes, the bacteria and archaea, you see the black spots, the black bar out there, you can see that their biomass is mostly concentrated in the subsurface, while compared to marine and terrestrial ecosystem. So these are very interesting facts. But when you think about our planet Earth, we think about green planet, right? Because a lot of photosynthesis is happening. and a lot of energy we are consuming from outside space. No matter is coming, but definitely a lot of solar energy is coming. And those energy are used for fixing carbon, for making food for other organisms. And any life on our planet are somehow related directly or indirectly. So that's our common notion. Like even if we do not photosynthesize, we depend on food products which are produced by primary producers who photosynthesize. But if you think about a dark environment, subsurface where there is no sunlight, it's a very extreme situation. It's an environment without light. It's oxygen deficient. It doesn't have a lot of nutrients, and definitely the conditions are pretty extreme in the subsurface. But still, we are saying that the largest reservoir of microbes are in the subsurface. So now we have to look at some of the facts and see that whether microbes can survive in those extreme conditions in the subsurface environment. So they are very like high temperatures, low oxygen concentration. There are a lot of nutrients which cannot be found there. But there was a recent study in China which reported that they found microbe in a deep borehole that is 5 kilometers from the surface. They found intact cells. So microbes can be found that way. And if you look at their temperature extremes, you can see they can tolerate up to a temperature of 130 degrees Celsius, way beyond temperature. And if we think about pressure, because we have got a huge amount of lithostatic pressure in the subsurface, even in the terrestrial, like if you think about terrestrial subsurface, we have got a huge amount of lithostatic pressure out there. But there are microbes which can withstand a pressure of around 1.5 gigapascal. That is around 15,000 times more than the atmospheric pressure. So you can imagine how much pressure they get. So the normal atmospheric pressure is 0.1 megapascals. If you just do your math, you can see that it's around 50,000. So it's huge. It's huge. So they can survive. But now they can survive those extreme conditions. But now we have to know that whether they can get their food and energy. Those are the main things for their survival, right? So we did a quick survey of different subsurface processes, which are pretty common. And we saw there are a couple of geological processes which can create a lot of chemicals which can act as electron donors and acceptors, which means that there will be a redox reaction out there from which we can gain energy, or from which any living being can gain energy. Such as radiolysis of water can give you a lot of hydrogen and stuff, and those can be used as electron donors. So there can be a lot of things that might be happening. And this can fuel a lot of different biological processes. And these biological processes will again create a lot of different products, which will be again utilized by different other biological processes. And some will get mineralized and deposited in those environments. Sorry. Sorry about that. So you cannot go back. I don't know why. Yeah, so if you look at these processes on the left, you can think this of as a rechargeable battery in our cell phones, in our laptops. So you can recharge, and you can again gain some products which different living beings can utilize. So it's kind of getting recharged by geothermal energy. Even if we know these facts, there are lots of questions which are unanswered, such as like what controls the subsurface microbial abundances? How are they distributed? What kind of microbial groups are there? What kind of metabolism they are performing? And how can cells survive in this exceedingly low energy fluxes? Because there is energy, but it's much lower than the solar energy. And one of the biggest questions is that how are microorganisms impacted by human activities? So we do not think about human activities which alter subsurface environment, right? But we do much more impact there. We are storing huge amount of nuclear waste. We are doing open cast mining. We are taking in a lot of gas, which is powering everything out here. Oil, coal, everything is coming from subsurface, right? And it is impacting the subsurface microbiome. But since we do not know who are there, what are they doing, we do not know their impact on us, right? So that is one of the questions which remains unanswered. So one of the studies that we tried to perform was in Deccan Traps, and this is a part of my PhD work. So Deccan Traps is one of the largest igneous province, which is located in the Indian subcontinent here. And if you compare to the Siberian traps back in the northern Russia, it looks much bigger, but since this is a 2D map, it looks bigger, but if you compare the sizes, they're pretty similar in size. So it's a huge province, igneous province, and it was formed around 66 million years ago. And a lot of scientists and geologists think that this was Deccan volcanism was responsible for extinction of dinosaurs. So that's still a debate, but it started getting creation. The volcanic activity started in a place called Reunion, and then there was continental drift, and the Indian subcontinent, where you see the Deccan Traps, started moving northwards towards Asia, and now you can see the Deccan Traps here. So we wanted to look at the subsurface Deccan Traps because there are two important features. One is definitely it's a igneous province. So in any igneous province, there is least biological impact from outside. So it didn't get metamorphosized or it didn't get transferred into something where you get a lot of organic carbon. So generally, you shouldn't find a lot of organic carbon there. And the second thing, if you think about astrobiology, there are a lot of other terrestrial planets where you can find this kind of igneous subsurface environments, right? So we wanted to look at that. So this is the green region out here is the Deccan Traps. And we selected a region on the Western Ghats. So Western Ghats are a mountain range in India with Martin. We selected a region out there. So this was not a random selection. There is a dam, a reservoir out there, which is famous for reservoir-induced seismicity. So that's why we chose the Koyana-Varna region. To give you a brief about background about reservoir-triggered seismicity, so it will give you a sense of why we selected those regions. So here, if there is a reservoir which is filled with water, so after heavy rainfall, what happens? The level of water in this reservoir increases, right? So when the water increases, the mass of the water increases, so it creates a pressure in the subsurface faults and fractures. So when they impounded the dam, they were not sure about the subsurface geomorphology and structure. They didn't know that whether there were a lot of fractures or not. So what happened after 1964 when the dam was impounded, they started seeing this small kind of earthquakes in those locations, which was not there before. And they found out that it was due to the changes in water level. So when it gets filled up, it will create pressure and create a effect of earthquake. And when the pressure is released, when they will release water, again, the mass will come down and again, the subsurface stress will try to come up and you will see earthquakes. So it's just like a balance. An old mechanical balance. So if you put on some stuff, it will go down. And if you put out the stuff, it will try to come up. And in both of these situations, you get seismic activities. And these seismic activities, we believe, like before drilling out there, we thought that it created a multiple faults and fractures in the subsurface environment, which are habitats or which could be habitats for different microbes, right? So that's why we chose them. So there were 10 drill sites as a part of ICDP project in Koyana-Varna region. And we chose 3 of them. Rather, it's better to say we got access to 3 of them. We were the last one who reached there because the first intention of this project was to install seismometers out there to get an early prediction. So they were not too much concerned about the subsurface microbiology. Then we went there, we started looking at the core samples and started collecting them. So first, when you drill in the Deccan Traps, first you will see basalts. Those are around 65 million years old. Then you will see a transition layer, which are weathered granite, and those might be exposed to outside conditions like before 66 million years ago. And then you will see granites, the basement rock, which is more than 2.5 billion years ago. So you will get a variety of conditions and variety of environments out there. But most of them are igneous in nature. So what we did, we started drilling through the basalts. We started collecting samples from 59 meters below the surface because we thought that the above 60 meter, even these environments are very low porous environment, but we thought that there might be organic carbon which might get penetrated from the surface to those regions. So we wanted to avoid that. We wanted to look at whether there is self-sustaining life in the subsurface without any photogenic input from the surface. We also looked at the groundwater aquifers because it's much more connected to other environments. And we try to look at their microbial habitats and try to compare like what are the things that might happen out there. So, sorry, these are some of the sites like Ukhalu and Panthani where we started our drilling, we collected our samples, and collection and curation of samples was one of the biggest goals for any for this project, for mainly for geomicrobiomes, because most of the samples when the geologists were collecting, they were getting contaminated with other stuff. Like because if you when you are pulling something up, there are a lot of chances like the drill marks, the overburden and other stuff can pollute that. So once we collected the rock samples, this step was very critical, very critical. So we used different dyes to look at like whether the drilling mud was penetrating into the inside part of it. So we were chiseling off, cutting with granite cutters, the outside layer, and we were taking the inner part where we couldn't see any dyes which were penetrating. So we were doing those kind of stuff. And in some cores, we did see where there were a lot of porosity or a lot of fractures, we did see that the dyes was penetrating to the core. So we threw them away. So we ended up with only 13 samples. Those were we called according to our characteristics pristine in nature. And then we did DNA extraction, and it was a real pain. It took me more than 1.5 years just to get simple DNA out of these rocks. And then we did sequencing, and we used different pipelines for looking at who are they and what they are doing. So we use 16S RNA gene sequencing for looking at who are they and what are they doing. We use 4-method genomic. We also use series of different geochemical analysis to find the environmental condition, to find the nutrient conditions, and find what kind of energy fluxes might be there in the subsurface environment. And with both the data in our hand, with the genomic and the geological data, geochemical data, we wanted to corroborate and try to reconstruct what might be happening. So first, the question is that whether Deccan subsurface is habitable. Whenever you go to any other place like Mars or other extraterrestrial planets, you first need to know whether it's habitable or not. So starting with the temperature as you go down, it increases 25 degrees Celsius per kilometer in Bassards and 15 degrees Celsius per kilometer in Granite. So when we reached the bottom of the drilling site, it was around 55 to 60 degrees Celsius, which is quite a good range for microbes. So we have seen a lot of microbes, thermophiles, to survive in those ranges. So we were sure that it is amicable for a lot of microbes. Then we look at the pressure. So it's 26.7 megapascal increasing stratospheric pressure. So even if we calculate that, it is kind of bearable by a lot of microbes. We looked at the pH. We didn't put a pH electrode in the rock. We provided them water. And we looked at the pH and wanted to see that in presence of water, what was the pH of that and whether any microbes can survive out there. And we saw that the range is pretty good for any microbes, for a lot of microbes to survive. And then we looked at biomarkers. Two of them, like Hopanoids and Diploptin, which gives us a sense that there were living beings or living stuff out there in the Deccan subsurface. So but most of the places we didn't find a lot of these biomarkers. A lot of these places we did find. But still, the biggest question remains. What's the biggest question? The biggest question is water, right? We need to know that whether there is water that's available in the Deccan subsurface. I'm not talking about the aquifers. I'm talking about the hard rocks in the Deccan subsurface, whether there's water. So we took help of a process called metasomatism. So metasomatism is the process in which what happens if there are faults and fractures, and in those faults and fractures there are certain minerals which get exposed. So these are original minerals. But if there is any fluid movements or hydrothermal water movements, what will happen is that this mineral will change in its composition. There will be modification in these minerals, which we call metasomatic minerals. And we tried looking at those metasomatic minerals in the Deccan subsurface, and we did find huge amount of metasomatic minerals out there. A lot of different metasomatic minerals, which gives us a sense that even we couldn't say, like we cannot say whether there is water movement now at present, but we can say that there were sometimes in the past, or it happened, water movements are there. So we looked at microbial diversity across those 13 samples because we collected a lot of samples, more than 50 samples, but according to the parameters that we set for aseptic for contamination-free samples, we ended up with only 13 samples out here. And DNA extraction was also a component out there. In some of the samples, we couldn't extract DNA. So here, the main take-home message is that if you look at basalts, the basalts have a lot more carbon, both in the form of organic and inorganic. And their microbial diversity is very different compared to the transition and the granites. And it corroborated with the metabolism that we found out. We did find a lot more heterotrophic members compared to autotrophs in the basalts compared to the transition of granites. So granites, we did find heterotrophs, but there were more number of autotrophic members which can fix their own carbon, but they were chemoautotrophs. So they were using chemicals for gaining their energy and fixing their carbon. Then we tried to figure out the overall carbon cycle in the Deccan subsurface. So it's a pretty normal thing, normal biotic process. But we try to figure out these red lines. These red lines denote carbon fixation. And all the carbon fixation can happen without the energy from the light. So they are dark carbon fixation. So we did find pathways in the Deccan subsurface where we found that microbes were using chemical energy. They were breaking their bonds, transferring that, they were electron donors and acceptors, which were responsible for getting those energy, and these microbes were utilizing those and fixing carbon. And there were certain pathways which could also fix bicarbonate, and we did find a lot of bicarbonate minerals in some layers, where we did find certain autotrophic carbon fixation pathways more than others. Then we try to compare the microbial diversity with groundwater and the surface water. And in the rocks, the microbial diversity was very, very different while compared to the groundwater and surface water. But the interesting thing was that when we tried to dissect the groundwater system, found a self-sustaining environment even in the groundwater that might be very different from the rocks. So it starts with fermentative organisms. So first we found fermentative organisms which were using dissolved organic carbon from the groundwater system, and they were producing carbon dioxide and hydrogen. The carbon dioxide were the input for autotrophs, and they were fixing those carbon. On the other hand, there were a lot of anaerobes which were using a lot of organic carbon and converting the electron donors and reducing it. So they formed a lot of converting the electron acceptors and reducing them. So they found a lot of electron donors after the anaerobic processes. So these carbon were further used by the fermentative organisms, the anaerobes, and also chemolithotrophs. So why I said chemolithotrophs out here, a lot of people get a bit anxious when they see that chemolithotrophs are using fixed carbon because they can produce also. But if you think about, there is a process called chemolithoheterotrophy, so which means that they can use the chemical energy. They can also use organic fixed carbon. So it's kind of a mixotrophic reaction where they can fix their own carbon, and also they can utilize carbon fixed by others. So these are kind of mixotrophs that we have found. So that's why I wrote just chemolithotrophs out. And a lot of hydrogen, they can also use a lot of hydrogen. There were hydrogen oxidizing bacteria, so it was used as an electron donor. And we saw some chemolithotrophs which were utilizing them for gaining energy. And the byproducts, and even these are again a lot of electron donors which are generated by that. They were further utilized by chemolithotrophs. We did see a lot of ammonia oxidizing, iron oxidizing, and sulfur oxidizing groups out there. And the byproduct of chemolithotrophy was again giving a lot of organic carbon in the subsurface system. And if you think about the minerals and other things that we located in the things, so we saw that in the subsurface setting of Deccan, we can get a lot of carbon dioxide and hydrogen from different geological processes. Since it's an igneous environment, there is always some kind of backgrounds of this hydrogen and carbon dioxide in those systems. And we also look at a lot of minerals like pyrites and others, which can get and release a lot of electron donors in this case, and a lot of microbes in those systems can utilize it. And there was a fascinating concept in the late 1990s where people thought of hydrogen-driven life. A lot of people didn't believe it, but we did find hydrogen-driven life. So even if you remove these geogenic sources, we will still get carbon dioxide, and remove carbon dioxide from the equation, just hydrogen, which can be a source of energy for the whole system. So it kind of supports the fact like deep subsurface environments are driven by hydrogen rather than any other products such as oxygen and other stuff. So to summarize this study, so we did find that Deccan subsurface is suitable for life, depending on different parameters and different electron donors and acceptors. The microbial community was governed by the geochemistry of the rocks. And there was definitely a huge interplay between the chemolithograph and the heterotrophic microorganisms, where there was a lot of exchanges of carbon which were going on. And distinct microbial communities were found in surface water and groundwater samples. This was very interesting because we thought that there were links, there were a lot of unconfined aquifers out there. We thought that the surface water were percolating in those groundwater environments, but we did find a totally distinct groundwater population out there in the Deccan subsurface. So this concludes the first part of my study, and I will be talking a bit about, as Francis mentioned, like about Antarctic microbiome. A lot of people get astonished that Avishek promised to talk about dark biosphere, but he's certainly talking about Antarctic microbiome. But I will try to convince you that there is also a dark biosphere. I'm not talking about the deep oceans, but also in surface oceans, there might be dark biosphere in the Antarctic. I will try to convince you. Yeah. So, yeah. Okay. So if you look at the Antarctic as a continent, it's a melting continent due to the climate change and other global warming and stuff. It's melting. It's a lot of ice covers in the Antarctic. And a lot of people, a lot of scientists are very curious about how it is impacting the world ecosystem out there, because there is a self-sustaining ecosystem out there also. You think about phytoplanktons, zooplanktons, nectars, there are fishes, a lot of penguins, a lot of things going on, but there are also a lot more to them. So just to give you a brief geography of Antarctic, so this is the west, this is the east Antarctic, this is the south pole, and we collected our sample from the Antarctic Peninsula, to be very precise, the western part of Antarctic Peninsula where the Palmer Station is located. So if you look at this figure from one of the NASA sources, you can see that the air temperature is increasing near the Antarctic, mainly the northern part of this peninsula. So this is a figure from 2020. And obviously we wanted, so one of the goals of the project was to see that how it is impacting the microbiome, or whether the microbiome in the Antarctic is crucial for the overall system, because there are lots of primary producers out there. There are lots of phytoplanktons who are fixing carbon. So it's quite like we do not need microbes out there. So that's the thing. Like we have a lot of algae and stuff which can produce, so we do not need bacteria and archaea, right? But we wanted to venture into those realms and wanted to see that whether they are part of this overall ecosystem. So these are the Palmer LTR grids as a part of like long-term ecological research. Each year, samples are collected across this grid, and we did collect samples from all these transects that you can see on the map. And we wanted to find out what kinds of microbiomes are there and whether they are critical for the overall functioning of the Antarctic ecosystem or marine Antarctic ecosystem. Now coming down to the basics of any trophic levels and trophic cascades, so you can see that in marine environment, phytoplanktons are the primary producers, a lot of algae out there. And then there are food for zooplanktons, which feeds on them and releases carbon dioxide. And again, zooplanktons are the food for nectars, right? So if there is a huge impact or huge climate change, the first thing that is going to affect are the phytoplanktons, because their growth and their different things are temperature dependent and also salinity dependent. If there is a lot of melting of water, the salinity of the ocean will change, which will create a lot of ripple out there, right? So if the first part is getting affected, the phytoplanktons, the whole thing will get affected because they are the primary producers. I'm not telling that their population will decrease or increase, because a lot of times we have seen due to higher temperatures, the population of certain phytoplankton increased on those systems. But it will definitely affect somehow or another. But if you think about the microbes, they are also part of this Antarctic ecosystem. How? A lot of these organic carbon and particulate organic matter are formed, which are utilized by a lot of heterotrophic bacteria in the Antarctic. They use them and they respire out carbon dioxide. And there are also microbial members in the fecal matters of these nectars and also zooplanktons, which can also decompose a lot of carbon. So they are also intricate part of this system. But there is an interesting concept of microbial loop where the organic matter goes to the bacterial members and again goes back up to the zooplanktons. So it's kind of a loop. The organic matter is not directly channelizing to the zooplankton, but it's going first to the heterotrophic bacterial members, and then heterotrophic bacterial members are food for different micro zooplanktons which feed on them. And even there are processes which converts this heterotrophic group converts certain carbon sources to recalcitrant carbon sources, which is not labile and which gets stored in the environment for long periods of time. And which we do not talk about often, viruses also play a very important role in the Antarctic process. How? Because they lyse the phytoplanktons and even other bacterial members, and they release a lot of organic carbon in the environment through two processes. One is like a viral front and one is viral shutdown. So I'm not going to talk about those. But what about 6 months of winter in the Antarctic? What happens there? So if you guys know, in polar region, there is negligible sunlight, but there is no sunlight during 6 months of winter, right? So definitely it will impact the phytoplankton community or population, and it will have a huge impact on the overall ecosystem because they need to survive for those 6 months. And a lot of things like a lot of nectars they need to feed on. They cannot store their food for a long time. There need to be food out there. So that was the driving thing, not for the project, but one of the things that I'm going to talk about today. So what we used a very simple methodology. We took the water sample, did DNA extraction, did whole metagenome sequencing, and look at the DNA sequences. And we developed a pipeline called Imagine, which does which filters the reads, sequence read, which assembles it, brings it, and reconstruct genomes from the whole metagenome sequences. So our main intention was to look at the genomic variability across different layers and also to get a sense that what might be happening in the Antarctic nights. But that was not the main aim of the project. And a disclaimer out here, we didn't collect samples during the Antarctic winter, but we are just looking at biosignatures out here. I want to be very clear about it. We are just looking at biosignatures from the summer community to get a sense like whether it can answer some of the questions in the winter, right? So we collected samples from different layers, and we did see that the genomic variability is much more in the surface condition, in the surface, which is the top 30 meters from or top 40 meters from the surface. And the deeper parts of the ocean, the genomic variability was much lower. So this is kind of pretty common because why? And kind of what we expected, because the surface layer is much more impacted by different meteorological conditions. There are much more changes, but the environmental conditions are much more stable in the deeper parts, right? So that's why we are looking at this, and we are saying that there is a lot more variability in the surface environment compared to the deeper parts. Now, I would like to give you a glimpse of the microbial ecosystem, and mainly with respect to the carbon cycle. So we did know that there were a lot of heterotrophic bacteria, but we were surprised to find that there were a lot more mixotrophic bacteria in the Antarctic surface while compared to the heterotrophic. And we were amazed. So here, what was happening is that a lot of phytoplanktons, which were not earlier seen in the Antarctic, now we're seen in the Antarctic, which has cellulose in the cell wall. And these heterotrophic groups, they had a wide array of polysaccharide degrading enzymes, we did find. So they were producing those enzymes, they were creating the labile carbon, and I'm talking about summer. And we hypothesized that those carbon are utilized by heterotrophic bacteria and the mixotrophes. And what about the deeper parts of the ocean where there is limited photosynthesis out there? So what we looked at in those regions is that the detritus or the fecal matter starts sinking as they form. And there are bacterial members which are found in both the shallower environment and also the deeper environments. And we think that they get attached to this detritus. And as they start sinking, they start releasing those labile carbon because they are decomposing it, mineralizing it, and also releasing carbon, which are further utilized by the heterotrophs residing in the deeper parts of the ocean. And we also did find different bacteriophages which were breaking, killing bacteria and releasing much more labile carbon. But the interesting fact fact was of the mixture cross. So this is all happening in summer. So mixture cross, as I mentioned, their metabolic lifestyle is very fascinating. I really like them. So if you think about cooking, right, at home, sometimes we really like cooking. Like when the weather is inclement outside, we don't want to go in rain, so we want to cook, right? When the weather is adverse. But sometimes on a fine sunny morning, we want to go to Starbucks or Jittery Joe's, sit outside with a cup of coffee and enjoy the weather, right? Somebody's cooking for us. Similarly, these guys have similar lifestyle. When it's adverse, they don't want to go outside and look for food. They will prepare their own food. They will fix their own garden. But if somebody is cooking for you, why will you spend your energy for cooking your own food, right? They do the same thing. So we believe, or I believe that in winter, this thing changes, this direction changes. They also start producing some level of carbon in the system. But it needs much more research for establishing these facts, because collecting samples in the coastal region in the winter in Antarctic is very challenging. But this is some of my thoughts, rather, I would say. And so to conclude, like depth plays definitely a very important role in microbial diversity. We did see a lot of polysaccharide degrading bacterial members. We were focusing on that, and we wanted to see that whether that can lead to a lot of labile carbon pool in the system. We did see a dominance of mixotroph in the surface layer compared to heterotrophs and autotrophs. We think that vertical product transport from the surface can be an important mechanism fueling the deep, dark Antarctic waters. And definitely, I didn't show it out here, but we did see that terrestrial ecosystem proximity. So there is a bird island out there, and we collect microbiome from the coastal region. And we did see that the microbial diversity is very, very different in this region. So the terrestrial ecosystem proximity and benthic fluxes, where it's shallow waters, the microbes are acting, or their metabolic potentials are very different from the open waters. So that also influences the microbial diversity. So with that, I would like to acknowledge a lot of my supervisors, my advisors, a lot of my colleagues, funding agencies, and a lot of people, a lot of labs whom I worked with, my lab members. But at the end, I would like to say one thing, that this field is like, if you think about aquatic life and deep subsurface, it's a growing field. It's just like a small plant. And I would suggest, like, if you get an opportunity to work in this field, you guys can contribute a lot, because we still do not know a lot about this deep subsurface environment or dark life in darker parts of the world. So I think we all can work together, contribute in this field, and move this field forward. With that, I would like to thank you all for your patience, and I will be very happy to answer any questions you have.